Okay guys, so let's look into some statistics, how these substitution matrices they are derived. So probably we would look into some probabilities and probabilistic models. So let's have a pair of sequences x and y. One of them has a length of n, the other one has m. So xi can be any symbol in x and yj can be the jth symbol in y. These symbols are derived from alphabets. So in case of DNA, the alphabets, they are coming from A, T, G, C, whereas the amino acids or alphabet size is 20, which can be any of those 20 amino acids. So what we want to do over here is we are given a pair of aligned sequences and we want to assign a score to the alignment that gives a measure of the relative likelihood that those two sequences are related as opposed to they are not related or they are unrelated. So we are trying to estimate the likelihoods of sequences being related versus non-related, and then we convert it into this score, which we put that score into these matrices. So we can have two models. So letter A occurs independently with the frequency of QA, and the probability of two sequences is the product of probability of each amino acid. So for example, we have on one side we have xi and on other side we have yj. So let's suppose if we have a random model, so we can have their independent probabilities and we can take the product of their probabilities and just multiply them. So here is the model where we can say that these sequences are unrelated, or they just pair up together by chance or by a random model. Whereas in the match model, we see that these aligned pairs, we are trying to correlate them. So here we are looking that it's not by chance, but actually they are related with one another. So here we can have their joint probability, A and B. So in that case, we have residue A and B, so that have been independently derived from some unknown original residue C in their common ancestors. So we are trying to say that these two residues, they are correlated with one another. So in this case, what we can over here is the probability of these two residues can be the product of the probabilities, their joint probabilities. So this is a match model. So we have P, X, and Y, while we are seeing it as a match model. So that is this sign over here, pi sign over here is the product sign. So that is the product over all values of I and the product of Xi and Yj. So we can take these two models and we can correlate with both of them with one another. So in this way we can come up with the likelihood. So we can have Pxy while M divided by Pxy while R. We put them together and then we put their values, and finally we are here. So that is our answer. So let's take their logarithms. So since we are taking these probabilities, they are really small numbers, they are less than 1, because you know that probabilities, they are taken from, they range from 0 to 1. So these small numbers, when their products are formed, we get really small numbers. We call it as underflow, or these kind of issues, we call them as underflow errors. So in order to avoid that, we take their log. So when we do logarithms, the products, they become sums. So we can get there those small numbers and convert them in on a log scale. We can come up with whole numbers. And since we are applying those logarithms, these values, they become their summations. So over here we have S, which is sum of S, Xi and Yj, where the score of A and B is actually the log of PAB divided by QA and QB, the same thing which we have done over here. So that is the log likelihood of AB as aligned versus unaligned pair, or log odd ratio. So using those log odd scores and substituting evolutionary time in it, these folks, Dayhoff, Shorts and Orcutt in 1978 presented their famous PAM matrices. PAM stands for point accepted mutations. Actually they use the substitution data from similar proteins and then use that whatever their results were to interpret into for those proteins which are having longer evolutionary distances. So here was the log odd score. So they incorporated evolutionary time in it. So, and in this way, these equations, they were a little bit changed, and they come up with these scores where we have that evolutionary time also. Then they get those values and rounded them into near integers and then scaled them. So in PAM 250, they scale to give the scores in third bits. So they take the log2 score and take 3 divided by this log2 score, whatever those log2 score was. So in this way, they were converted into these what we call them as third bit scores. What the issue with the Dayhoff matrices was that since they were using the closely related proteins, the similar amino acids, and while they were interpreting those for longer evolutionary distances, but later on it proved that they were not performing well in case of those distantly related proteins. So Henikoff and Henikoff in 1992, they came up with another scheme. We call it as Blossom. These are Blossom matrices. So they are actually derived from set of aligned ungapped regions from protein families. We call those families as blocks. So actually within those proteins, we have some regions or some conserved sections. We call those as families. So these are actually those secondary structures and then tertiary structures in the proteins. We are not going into the details. So we have families and superfamilies and folds and blocks. So these are the structural units within those proteins. So those regions, they were called as blocks. So they what they have done was they take those sequences coming from different blocks or which are present in which are sharing those blocks, and then did some clustering analysis. So with those clusters, they gave some scores to those clusters and retained those clusters that were having some similarity scores above some threshold. We call it as L percent or some percentages of that. Those members within the clusters were that much percent or more than that similar to one another. So let's look into this Blossom 62. Why 62? Because the score or similarity score is above 62%. So the clusters are highly related. So their similarity is no less than 62%. It can be equal to or it can be greater. So here we have A. Substituting an A, the score is 4. A goes with D, the score is -2, and so on. So in this way, they came up with this matrix. We call it as Blossom 62 matrix. So substitution scores, they can be derived from probabilistic models, and PAM and Blossom are some famous substitution matrices.